Thank you again. It's a pleasure to present this lecture to you. In the two previous lectures, we've discussed the role of the oocyte in both normal fertilization and abnormal fertilization. And so what I'd like to cover in this lecture is where do we really stand in terms of understanding what the features of the oocyte are that could predict the quality of embryos made from those oocytes. And as you'll see, I think there's been a great desire to be able to bring some meaningful measurements of oocyte quality into the field of human ARTs, but to date, we've had limited success. And this is one of the reasons. The production of a viable oocyte is a function of its developmental continuum. There is a long duration. In the human, we estimate this to be in the order of 100 days of intra-ovarian development, where the stages of meiosis are essentially blocked at diplotin, and yet gene activation, transcription, and translation of important maternal products have to take place during a protracted growth phase of oogenesis. There are very significant changes in the imprint marks within the female or maternal genome over this course of time. And as we mentioned, it takes on the order of 100 days to sustain all of these processes, including the acquisition of the ability to complete meiosis and produce an embryo. So when it comes time to triggering ovulation, we really have to rely upon the final quality checkpoints, if you will, that will take the ovulated oocyte and prepare it for fertilization and subsequent development. Because embryogenesis, right at the start of development with fertilization, is an energy-demanding process. It requires extreme parameters of polarity, meaning where things are is important, as we've mentioned, and involves major regulatory changes in cell cycle progression and the genomes themselves. Maternal and paternal genomes themselves have to undergo significant remodeling in order to support and sustain development. So our outline for today is to see exactly what has been accomplished in the way of predicting oocyte quality in human ARTs. And as I mentioned, it remains a challenge. So we'll look at exactly why this has been the case. Again, we have to reflect upon the fact that building an oocyte is a long, carefully regulated process that is very complicated. And I'll review for you some of the features that we've come to understand, especially now in the context of being able to study human oocytes directly because of the efforts of the fertility preservation movement. At the heart of this is the fact that the quality of the oocytes that result from oogenesis is directly related to the qualities of the follicle. These are integrated and inseparable processes, and in our daily practice of ARTs, we are likely trying to separate these two to the detriment of the oocyte quality. And that's why you'll see that the field as it stands now has realized that our chances of evaluating oocyte quality directly by monitoring some dimension of the oocyte's physiology, etc., are really limited, and that we're turning our attention to evaluating both the gene expression and physiology of the cumulus cell in order to get a better picture of what the oocyte's quality means. We don't have many options, okay? Again, we strive to achieve as many mature oocytes as possible with our technology. We know that age is a dominant factor in terms of not only the declining numbers of follicles with advancing maternal age, but we also know the quality of oocytes from older women make for difficulties in producing embryos and babies at the end of the day. And we have to keep in mind that, ovarian physiology dictates that most of the oocytes in the ovary will die. Most will be eliminated. Very, very few will make it to the stage of a Graafian follicle where they can be successfully ovulated. And processes are now being understood. Of course, apoptosis has been on the center stage for some time as a determinant in follicle health, ovarian health, as has necrosis and immune conditions. But we're seeing now that this field of autophagy, which is a basic cell survival mechanism, is one that needs to be explored. Now, finally, the other complicating factor here is that there are many confounders. We'll mention a little bit about patients in line for fertility preservation, but we're seeing more and more that environmental toxins are interfering with ovarian function and therefore egg quality, as is lifestyle. So it's a tough problem to resolve. So the question is, why is it so difficult to move forward in this field of oocyte quality assessment? First thing I think we have to keep in mind is that looks can be deceiving. Okay, we have especially when we transitioned from true IVF, retrieving cumulus masses and inseminating them directly, when we transitioned to ICSI in the 1990s, we at least gained the ability to more directly evaluate morphological parameters of the oocyte in relation to the appearance of the polar bodies, okay, and other parameters of zona integrity, cumulus corona. But, as we all recall and see on a daily basis, not all follicles respond to controlled ovarian stimulation and triggering the same way. And so while we're likely to pick up these beautiful, mature, expanded cumulus masses for ICSI at the end of the day, we just don't know a lot more about the quality of the oocyte other than the fact that it's probably undergone nuclear maturation. And that's part of the problem, because we've now seen that oocyte quality is not only dependent on the level of the nucleus and its nuclear maturation or completion of meiosis, but there are a huge number of cytoplasmic and membrane changes, plasma membrane changes, that have to occur if not only fertilization is to proceed normally, but even later development. And the punchline for this lecture is that it does seem to be the metabolism of the follicle and the cumulus oocyte complex that dictates the fertilization and developmental competence of human eggs. So, not so easy to get a handle on, but it does appear that the biological problem that we're dealing with is it takes a long time to make a good egg, and it's complicated. So here are two ways of looking at this, the repertoire of factors that go into building an oocyte initially, and then maintaining its interactions with the follicles over the long period of folliculogenesis. So here's a breakdown from a review article we published some years ago of the just an incredible communication network that exists between the oocyte, its cumulus cells, follicle cells, and even the theca. Dictated, of course, not only by the actions of gonadotropins, but steroids, and dictated, as we have come to learn, by factors that are expressed, genetically expressed in the oocytes themselves, that impact many aspects of the metabolism of the granulosa cells. And of course, this is going to extend not only from the initial activation stages of the follicle, but now, for example, if we just look at ovulation, there are any number of genes that have now been implicated in the process of oocyte maturation, really, that come from the oocyte or the granulosa cells that are essential for building an egg. And then if we look over here, we can see that while we initially thought that there were just these simple interconnections via gap junctions that could share the metabolism of the somatic cell with the oocyte, now we've come to understand that there are many other pathways, signaling pathways, involved in this dialogue that really determines which eggs will acquire all of the properties needed for development and which eggs will not. So this unifying theme that we and others have built on is that the achieving high levels of oocyte quality is really a reflection of the metabolic support received not only during the growth phase of oogenesis, but especially during the period of time at ovulation and after triggering. So if we take our crude viewpoint of morphology, as I said, we imply nuclear maturation simply by seeing a polar body. Of course, the pole scope, when it was introduced some years ago, also gave a very reliable measure of spindle integrity, which was good to have on hand, especially when, as at the dawning of ICSI. And some other efforts using the pole scope and other metabolomics tools, have tried to add to the repertoire of things that we could measure. But, I think the main focus has always been on the genetics. Did the meiotic cell cycle resume and progress at a normal rate to metaphase 2, yielding a spindle in the metaphase 2 stage that had essentially the requirements upon fertilization to exercise release of the second polar body and move towards activation of the egg's metabolism. Now, again, this is about being in the right place at the right time. So the cell cycle control, which modifies first and foremost the regulatory proteins that control cell cycle progression, they control the stability of cyclins, they control the degradation of cyclins, the CMOS types of components. These are all very high energy consuming reactions that involve, in approaching metaphase arrest, the hyperphosphorylation of many proteins, and in leaving that state with emission of the second polar body and egg activation, the dephosphorylation of these. And at the very outset, there are dramatic changes in the organization of the cytoskeleton, okay? This is an image of an immature human oocyte. Here is its GV. You can see it has a cortex made of actin stained in red. And here you can see all of the microtubules that in fact will be used not only to build that meiotic spindle, but they'll be used to build all of the mitotic spindles in the embryo. And here you see a color and black and white image of the actin network of a human oocyte in metaphase 2. And you can see the spindle domains over here. And if you recall the previous lecture, the asymmetry in this filamentous organization, which is enhanced for your viewing pleasure over here. So here's the assembly of a huge network of actin in the cortex, and you can see these filament poor cortical regions and this central dense region. So this is an asymmetry that consumes a lot of energy, okay? And that energy has to be derived during ovulation and oocyte maturation. And what it has led to is, an important paradigm in which the various gonadotropin-mediated reactions in the granulosa cells increase. For example, an anti-inflammatory environment. They motivate most of the somatic cells to begin to move and reorganize, okay? And they regulate the cell communication between these cumulus cells and the oocyte. And it's actually now very well studied. This is a paper from some years ago that really demonstrated that the developmental competence of an oocyte that received appropriate FSH priming at the later stages of follicular growth, okay, produced a much higher percentage of high-quality embryos than those in which the communication pathways were compromised. And it's in many cases now been identified that this is not only nutritional molecule transfer from the cumulus cells into the oocyte, but also bioenergy substrates as well, such as ATP and GTP and glutathione, which we mentioned before. So this really has set the stage for the attention of most investigators now to look to the transcriptomic signature of the cumulus cells to predict the developmental competence of an enclosed oocyte. Now, that said, it's already become insufficient to study the transcriptome alone. So that other of the omic technologies are being introduced and applied to assessing, again, the quality of the somatic cells to gauge, the quality of the oocyte. Because gene expression alone, while it is significant to integrating some of these pathways, gene expression alone does not identify the proteins that are highly relevant or their functions. And there are many, many regulatory functions that these proteins are going to serve. And we need to identify those if we're ever going to come up with promising directions for how to gain more knowledge, more direct knowledge about oocyte quality. And at the heart of this is much to do with the management of chromatin during development. This is a very high magnification image of the chromatin status in a small growing mouse oocyte versus a fully grown one. These are human oocytes in the primordial follicle stage, giving you a nice representation of their diplotin meiotic condition. And here's an electron micrograph of a fully grown human oocyte with its prominent nucleolus. And these are all conditions that we know can be, again, affected by lifestyle. You can imagine that interruption of gene expression patterns during development of an oocyte could have long-term consequences when that oocyte is ovulated. So a couple of reflections here. Why is it that, especially in human eggs, we see that they're in this precarious situation, being stored in the ovary as primordial follicles for years and years. We're coming to recognize that even in the quiescent, so-called quiescent primordial follicle state, there are dynamic changes going on, and certainly the metabolism of the follicle is imposing an influence at the very earliest stages of the follicle. And, we know that many of these oocytes can make it through the growth and maturation phases. Certainly the way we do controlled ovarian stimulation is intended to rescue or maintain follicles that most likely would have been subject to atresia. And when we retrieve oocytes from these kinds of follicles and stimulation regimens, we know they're capable of initiating development. They seem to be able to undergo fertilization and cleavage, but they're unable to establish a term pregnancy. Again, the vast majority of the oocytes that are fertilized means that we're posed with a serious problem in terms of identifying high-quality oocytes. So human-assisted reproductive technologies, in essence, enables the collection of more oocytes, but they're not better. The research into the human condition has been stimulated in many ways with the advent of fertility preservation technologies, and as some of you may know, where a piece of ovarian tissue can be brought all the way through to a mature stage through this multi-step procedure that has been advanced and refined by Evelyn Telfer and Richard Anderson's group in Edinburgh. And it's really bringing us back to the point of understanding that oogenesis and what makes a good oocyte support embryogenesis is really the result of a complex interplay between ovarian physiology and, of course, reproductive tract physiology. That we're seeing that epigenetics is applied universally now in gamete production and embryo transformations, that the capabilities of the oocyte rely upon this maternal inheritance. And we're seeing, I think, a paradigm shift from the use of genomics technology, which is an important front end, to that of cellulomics and metabolomics. We need to keep in mind that sperm are contributing more than we previously suspected. And again, this is a moving target based on lifestyle susceptibilities. So I'm going to close by reminding everyone that the various competencies that an oocyte acquires, are a function of these various developmental landmarks, and that again, embryonic development is readily compromised by imperfections during the process of oocyte maturation. I think is a key to our future understanding. Thank you. Please keep your eyes open and exercise caution as we move forward, and there's a lot to look forward to. Again, I acknowledge my various colleagues over the years and the patients who have given us the materials to really study these challenging problems. Thank you.